and is in the throes of developing a multi-arm, multi-stage trial platform. She was appointed National Specialty Lead for Neurodegenerative Diseases in 2017 for the NIHR here in the UK. And actually, Camille is not with us today, so Simon and I had an opportunity to pre-record a session with her, partly because she's at an NIHR meeting just today. We had the most illuminating discussion about the state of research and how we are going to get research restarted here in the UK. So without further ado, I'm going to ask Eros to show the film. Thank you very much, Eros. Please do bear with us. We're just having a momentary glitch in sharing the screen.
The answer to how research in the UK has been affected by COVID is almost a tale of two cities, actually. So on the one hand, the UK has done brilliantly in research delivery, of course, through delivering the COVID studies. And that's really demonstrated what the UK is capable of as a research delivery infrastructure. The platform trials, the site engagement, the patient engagement at sites, the follow-up capacity, all of that is a really good demonstration of what we can achieve within the UK in terms of delivering clinical research. But of course, the flip side of that is that a lot of other research wasn't able to be sustained for a variety of reasons and different reasons across the year. Excuse me. So at the beginning of the first wave, one of the main impacts was losing staff to delivering frontline care or being deployed to other clinical areas where care was required. And then losing research staff also to delivery of the COVID studies, the urgent public health studies investigating treatments for COVID. And that was hugely impactful right at the very start. There was the sort of COVID lull that we went through in the summer and early autumn, where we were able to the staff returned to their normal research roles in part, not completely, but in part. And that allowed us to think about restarting some of our studies. And I'll come back to how we try to facilitate that as a network and as a specialty area. And then the very good news was that subsequent to that were the vaccine studies, as vaccines were being developed and then rolled out. So that meant that there was again a drain on staff, a pull on staff away from non-COVID research to deliver the vaccine studies. And of course, we're reaping the benefits of that, but it does mean that other aspects of research delivery have suffered. And what that means in terms of the UK and its research delivery position globally, I guess, isn't entirely clear, but there are certainly some risks that have become apparent that we need to be alert to and mitigate if we can. And an important aspect of looking at the risks is to think about who the stakeholders are who are disenfranchised as a result of this past year. And an important group of stakeholders are people living with Parkinson's and their care partners who haven't had the opportunity to participate in research. But an equally important group of stakeholders are the people who want research delivered. And to a large extent, that's pharma, it's the drug companies developing therapeutics that people with Parkinson's can benefit from. And we absolutely need to make sure that as a country and as a research network and as sites and as people with Parkinson's, that we are poised and ready to deliver studies for drug companies, because we want them to be delivered in the UK. We want the UK to be a vibrant, effective and sustainable place to deliver research for drug companies. because that's our lifeblood and also where a lot of the therapeutics are going to come from. But another very important group of stakeholders are funders based within the UK, government funding streams and charitable funders such as yourselves, Cure Parkinson's and Parkinson'S UK, and some studies that might be funded by external charitable agencies such as Michael J. Fox. These studies have also suffered, and of course the funding capacity within the charitable sector has suffered because the fundraising capabilities haven't been as they were. And then another group of important stakeholders are the research delivery workforce. And by this, I don't mean people employed to deliver research, such as research delivery nurses at sites or research practitioners. I mean, the investigators of the future are academic trainees, people doing PhDs or postdoctoral fellowships, or new investigators who will have lost the opportunity to engage in research. They might be struggling to gain their higher qualifications, or some of the experiences that they would have hopefully achieved over the past year. And it's really important that we put processes in place to mitigate the impact that they have felt, that we do have clinical researchers for the future.
Camille, how have you found how has the process adapted to this new world, the process of running a clinical trial?
So in truth, if it were just one new world, it would be easier to adapt processes, but it seems it's a new and different world every week almost. And there certainly were periods of last year where we got processes in place, and a fortnight later the world had changed yet again, and those processes had to be adapted. So we think we have got some processes which are new and which hopefully will be able to sustain moving forward, and then some which are no longer needed, thankfully. But in general terms, there are challenges if we think about how we're going to deliver research in the new world, whatever that looks like, and that of course isn't clear. Compared with how we were delivering research at the end of 2019 and the first part of 2020. So, and we can think about the trial journey. So if we are looking for sites to set up studies in, sponsors or research organisations or companies might look at the UK and look at the sites where they have traditionally engaged with and see whether they want their studies to be placed in those sites or whether other sites might be a good place to base those studies. And on the whole, that was done by reputation and experience. And now, of course, those sites might be the very sites, at least they were certainly in the first wave, that were hardest hit by COVID. Those sites might be much busier delivering COVID vaccine studies or other urgent public health studies, and maybe those sites will have less capacity to deliver Parkinson's studies. And we can be a bit more intelligent now, given the way that we utilise data and the ability to utilise data that has grown out of COVID and the research network's response to COVID, which has been very much driven by data and using it in order to ensure that the COVID studies are delivered. We can use those same processes for seeing how other studies are delivered in other disease areas, such as in Parkinson's. So we can choose sites mapped to patient need. We can place sites where the greatest burden of disease is, or where there are less competing studies. And by competing studies, I don't mean for patients. Very few studies actually compete for patients. There are plenty of patients. The competition is for the staff resource and the space at site, and maybe pharmacy capacity or capacity of some of the other services, such as imaging. That's where competition lies for sites in order to deliver studies. So we can be more strategic and intelligent about where we place studies. Once we've made that decision, we can think about how to find patients, and ordinarily we'd have face-to-face clinics. We might have a research nurse in the clinic. Excuse me, if we're particularly lucky, that research nurse might have their own room, so we can have patients that we're seeing in a consultation and say, would you mind seeing the research nurse next door? If they've got sufficient time left on their parking, they might be happy to go and have that conversation. And that's a very easy way of getting patients into studies, and it worked well. And the advantage of that, of course, it's the clinical team having that research conversation. And we know that when a clinician makes that research approach that's beneficial for patients. They value and trust the opinion of their clinician to guide them towards research opportunities. Now we aren't running face-to-face clinics on the whole, or at least we haven't been for most of the last year, although of course we're starting to restart those clinics. The patient pathway is different, and so it should be different because let's face it, the patient pathway wasn't set up with the convenience of patients in mind. It was set up for the convenience of the healthcare organisations. So it is absolutely appropriate that we think about how we design our clinical service to better map to the needs and realities and necessities of patients and how they want and need their care to be delivered. And within that, we need to think, well, how now are we going to offer these research opportunities? Because our traditional method of so doing will not be fit for purpose, or it just won't work because it won't be there. So we do need to do some work around that. And we can use data analytics again to help us, although that does take out some of the personal element, which is important for recruitment. So I think that we're in a transition phase where we're trying to take from the old and transition it into the new. We're not quite sure what that looks like because, of course, our patient engagement processes and tools are actually quite, I'm going to say Stone Age, but I mean pen and paper, so I'm guessing that's not Stone Age. But, you know, a participant information sheet in hard copy thrust in your hand, or that arrives in the post, or worse, as a PDF in an email attachment, that isn't going to be as engaging or as informative as an animation that you can click through on YouTube or something. Yeah, so the way that we utilize the real power of the digital technology that we have available is not maximized by a long stretch. So there is a lot that we can do to engage patients in research in a different way, to replace the face-to-face clinic consultation that may or may not happen in the future. And for a lot of studies, and particularly decentralized studies that are delivered completely remotely, which you know will be the future for a large portion of studies moving forward, we need to think about how we do that patient engagement so that it's a pleasant and fulfilling experience for patients to participate in studies right from the first moment that they hear that study name and click that first link on the internet. And having, as I say, having a PDF that you can download, I'm hoping we can do a lot better than that.
And that pause, restart, pause element of the lockdown, how has that affected screening and recruitment within the context of clinical trials?
Yeah, so I think in the Parkinson's portfolio, it has hit some studies hard for sure, and others not so much. So what we undertook in Parkinson's disease, which was similar to a process that was undertaken by the motor neuron disease community in June of last year, appreciating that there was going to be an opportunity for restarting research, albeit in a probably time-limited way, which of course turned out to be the case, was a process for giving guidance to sites about how they might wish to think about which studies to open and in what way. And this is on the back of 3 months of very hard work, actually, between COVID starting to impact in March and then being in that position in June. And what I mean by that is that the research network alert to the pressures on studies that were not COVID related, created a restart framework for sites to think about how you might approach reopening the portfolio at sites, and study sponsors, study teams, and funders, and indeed the regulators also on board with thinking, how can we deliver as much of the portfolio remotely as is possible, or what aspects of trial delivery can we convert to a remote format. So within Parkinson's disease, we grouped together some members of the national specialty groups. These are clinicians, regional leads for delivering Parkinson's research across the country, together with people living with Parkinson's, yourselves as representatives of charitable funders, and looked through all of the studies that were open to recruitment that had been paused as a result of that COVID first wave, and think about how important is it for us as a trial community, Parkinson's trial community within the UK, to deliver this study now. And we decided some rules that we felt were appropriate to suggest that a study ought to be targeted or prioritised. These were studies that allowed access to therapies that otherwise would not have been available to patients, such as the 24-hour subcutaneous levodopa infusion studies or neuroprotective studies. These are not therapies that are available to patients outside the context of a clinical trial. So we wanted sites to concentrate on those. We were also keen to identify studies that were close to hitting their recruitment targets, because of course they could be finished and primary outcome realised, either positive or negative. So it was important to concentrate on those. We also thought that for study delivery sites, it would be helpful if they knew which sites, which studies rather, could be delivered more remotely than others, because at that time in June, July, August of last year, quite a lot of the research staff were still deployed to delivering vaccine COVID studies, even though they might not have been needed for care delivery. So the research team wasn't what it was, and so studies that were easier to deliver, maybe not requiring such a specialist skill set, maybe that could be delivered remotely, are more likely to be delivered successfully. And of course, with the Parkinson's population primarily being an older population, and of course not vaccinated at that time, there was much less enthusiasm for coming to a central site such as a hospital or a research clinic because of concerns about COVID. So we set out a guidance document or advice, thoughts about how we thought studies could be delivered most easily within sites, and that actually has stood the test of time for the Parkinson's portfolio. A few studies have slipped, between those grades, particularly studies that were paused completely and then have reopened. So at the time that we undertook the exercise, they might have been paused, and now they might have a raft of remote delivery solutions in place which would allow them to be delivered more successfully. We had a window in the autumn of last year, August, September, October, before the second wave hit, where we were able to open studies successfully to recruitment again, and we recruited to studies such as exenatide and other studies which were important to allow access to therapeutics for patients. And then, of course, we had to pause those again over the winter. So it has been a bit of a zigzag, as you alluded to.
You talked about remote delivery tools. Have you got some examples of things that have been put in place that will enable trials to be delivered more remotely, more decentralised?
Yes, indeed. We undertook a project last year within the CRN to look at aspects of remote trial delivery, appreciating that this was a need for study delivery teams at sites, and also for investigators and sponsors to think about how their trials could be adapted to make sure that they were sort of COVID resilient and sustainable. And so we convened the remote trial delivery working group with a range of stakeholders and undertook a couple of surveys. One was for the professionals, people involved in delivering studies and designing studies, and another survey was with trial participants and their care partners to understand what has been put in place so that we could share that just to prevent people from reinventing the wheel, because of course a lot of processes have been put in place, and studies across disease areas have been trying to design solutions for the same challenges, such as remote consent, or how to get drug out to a patient, or how to conduct a study visit remotely. And beyond that, of course, there are all the sort of the infrastructure and processes around trials, which are based on face-to-face in-person contact. So things like ensuring that oversight is maintained by the principal investigator at a site, ensuring that data integrity is maintained, where the source documentation is, making sure that delegation logs are up to date, that monitoring can take place. For all of these bits of trial delivery, some of which are evident to the participants and some of which not, there have to be remote processes in place. There have had to be remote processes developed, and they exist. And within this space, there's quite a lot of guidance that has been created by several groups, including our remote trial delivery working group, that people can access. So on the Google site that we've created, that there is guidance around the enablers, the pitfalls, the challenges, the benefits of undertaking these processes remotely. And the sorts of questions that, if you're faced with delivering a study remotely as a study designer or a study deliverer, the sorts of things that you might want to think about, and then a link to resources that address exactly what you asked, Helen, about examples. So where e-consent has been used, or where telephone consent has been used, and where a remote monitoring solution might be in place, that sort of thing.
Yeah, it does seem that there are a plethora of these tools that are being developed, and I think having a central resource for them as sort of central hub for them, so that it makes it easy for people designing studies to actually pick out the tools that are going to be right for them, for their study, feels like.
Yeah, and actually the people that have put most work into this are the people who've been at it the longest, and that of course isn't our remote trial delivery working group, which convened last autumn. It's the people that were thinking quite far ahead of the game, and thinking about how this can improve the trial experience for study teams and participants, even before COVID.
Just playing a devil's advocate a little bit here, you know, clearly some of the feedback that we've had in the past is one of the reasons people take part in clinical trials is because they're part of something important and something that could make a difference. And some of the benefit, I think, for individuals has focused around actually going into clinic and discussing the trial with, you know, the knowledgeable study team who are leading it. And with remote delivery, I think that does become slightly diluted. So I think it's something I suppose I've been thinking about quite a lot, is how to make people feel really involved and really, you know, reflect that commitment in an important way, actually. So trying to think of tools to improve the communication in that way.
Yeah, I think there are three elements to that I'm going to pick up on, and two of which you mentioned. So I'm going to start with the third because I didn't write it down and I'll forget it otherwise. And that is the better clinical outcomes. So we know that people who participate in research studies on the whole have better clinical outcomes than those that don't, over and above the healthcare organisations who are research active on the whole have better clinical outcomes than those that aren't. So I've been thinking exactly the same thing as you, but from a slightly different perspective, is to think, well, what is it about that research experience that enables people taking part in research to have a better clinical outcome? Is it the face-to-face contact, or is it just the sense of contribution? We don't actually know what it is, but it is important that we try and understand what it is to ensure that we don't lose that if we move to a completely remote trial delivery experience, or even a partially remote trial experience. We don't know what the baby is in this particular tub of bathwater, but we certainly don't want to throw it out. And particularly because this is a key driver for services and for organisations to become research active, if we are able to demonstrate, which we have been able to do very robustly, that patients under their care will have better clinical outcomes if they follow this path of becoming research active. The two elements that you talked about are really important, and it might be, of course, that it's these two elements which contribute to that third, and it's that sense of involvement and that sense of feeling valued. And I think there is work to be done there. I think both of those things can be achieved for sure remotely, but it does need some thought to be given to it. That not every face-to-face interaction results in someone feeling valued for that particular contribution. It can go wrong, and it's the same for remote. It doesn't have to be a devaluing an experience. It can be done well. So as long as we understand what it is about what works and what doesn't work in whatever modality of interaction that we choose to employ, then we can harness those benefits. But that does mean that we'll have to think about things differently and make sure that patients aren't disenfranchised if they aren't able to avail themselves of a particular modality that's being employed. So, for example, if they don't have a webcam, for example, built into the device desktop that they're using, they won't be able to engage in a Zoom conversation in the same way as we are now. And so that might be limiting for them. They won't be able to get that same sense of personal connection if other people aren't able to see them. So I think that's worth thinking about. It's worth thinking about how the solutions could be put in place. And it might mean that that determines, is part of the decision-making process for people participating in studies in a way that it wasn't in the past. So if somebody does have a preference for an in-person study delivery mechanism compared with the remote, or vice versa, that might factor into which studies they choose to participate in.
Camille, have patient views, not their needs, but their views, been part of the adaptation process and the reopening process?
I mean, that's what you're describing, that reopening process, to some extent happens on a study-by-study basis, and the mechanisms in place for gaining those views will differ study by study. Most studies these days, particularly, of course, NIHR funded studies or charity funded studies, we would hope would have a patient representation on the trial management group who absolutely would feed into the restart process and how that can be done to maximise the benefit for patients. In terms of the overall structure of how things can work, as I say, we did have patient input into the advice that we provided for sites, and we also had patient input into the remote trial delivery guidance, very much asking patients for what and their care partners what worked well, what didn't work well, and use that to inform the guidance that we've put on the website.
That was the most amazing insights from Camille in terms of some of the challenges that research has faced in the last year. And please, the most important thing is if you have any questions, do use the Q&A function on the screen at the bottom of your screen, and we'll do our very best to answer as many questions as we can. And if we can't answer them live, then we will, during the Q&A session, we will try and post you answers afterwards. I am delighted to now actually introduce you to our research manager, Dr. Lea Merceline. Lea's been a member of the team for, I think, 8 years now, unbelievably. And it's fantastic to have her back from maternity leave. Lea's been having a look at our own research portfolio and how that has been impacted by COVID. So having had Camille's general introduction of the state of research and the massive challenges that the research fields have faced, it'd be great to hear from Lea about our funded work. Lea, over to you.
Thanks, Helen, and thanks, Camille, for that really helpful overview of how clinical research has been impacted, particularly in the UK. I'm just going to share my screen. I only have one slide, so I'll keep it brief so we can move on to the next talks. So I hope you can all see this. So Cure Parkinson's research has also been impacted by COVID-19, particularly the clinical studies that have been ongoing within the NHS, because of the recruitment suspension that Camille mentioned in her talk, and because clinical researchers were redirected to frontline services. However, the good news is that Cure Parkinson's clinical studies have now resumed, pretty much all of them have now resumed recruitment, and many of them were also able to continue throughout the pandemic using remote assessments like Camille mentioned. So you can see in this diagram, it's just an overview of our funded clinical research and where they are in terms of getting back up and running. So you can see most of our studies are under this category where they were delayed because of recruitment, but have now resumed. So here we have the exenatide sub-studies, the brain imaging and wearables technology sub-study of the phase 3 trial of the diabetes drug exenatide. This is now recruiting, and it's recruiting really well, and most of the sites are now up and running. We have the ADePT-PD sub-study, which is focused on a drug which is used for depression called nortriptyline, and whether it can potentially slow the progression of Parkinson's disease. This study is now recruiting as well, and has been able to set up remote assessments for their protocol to try and prevent any further delays. Professor Annette Schrag will be talking soon, and will give a lot more information on this study. And then we have PD-Frontline, which is a bit different in that it wasn't delayed due to recruitment. Recruitment continued throughout the pandemic, but there was a slight delay because this study was looking to identify people with what's known as the GBA mutation of Parkinson's. And as part of that study, participants had to send in saliva samples for genetic analysis. And during the first lockdown, this process of sending in these samples wasn't allowed to take place. So the good news is that's back up and running. That started again in September and is still recruiting. So you can sign up for that, and you'll hear more about that from Professor Anthony Schapira in his talk a bit later on. And then we have the azathioprine sub-study, which is a trial of the immunosuppression drug azathioprine for Parkinson's. This study was about to start before the pandemic broke in March 2020, but has now started as of the 12th of April. So recruitment is now taking place. We're really pleased about that. The two studies here are actually much less delayed because they were able to continue with their study visits. And as Camille mentioned, they were very close to finishing their recruitment. So these two studies weren't based in the UK. Lixisenatide is in France, and liraglutide is in Los Angeles. And although the very last participants did take a bit longer to recruit because of the pandemic, they have now completed that recruitment, and the study visits are ongoing. So we don't foresee there to be many more delays to these studies, and hopefully we can get the outcomes to the dates that are planned. And these two drugs are both diabetes drugs like exenatide that are being tested in phase 2 trial for Parkinson's. And finally, these three studies here of simvastatin, which is a drug used to lower cholesterol, that was tested in Parkinson's, UDCA, which is a drug to treat liver disease and gallstones, and the TransEuro follow-up study, which was a study doing long-term follow-up on participants who were involved in a stem cell transplant trial. These studies were able to quickly switch to remote assessment. So the delays were quite minor in these studies, which was great. And simvastatin was able to complete during this pandemic. And UDCA has now finished all the recruitment and all of the study visits for all participants. And so the team is now just doing the analysis of the results. And the TransEuro study is continuing the follow-up of the patients, and should be continuing for another few years. They just need to do some brain imaging, which is obviously in person rather than remote assessments. And this can now take place, which is great. As for our lab-based research, which I haven't listed here, that has actually been able to continue quite well during the pandemic, particularly from last summer, because most universities in the UK and overseas were able to reopen last summer and stay open with COVID secure measures being put in place to ensure that lab-based research continued. So although it has been a really challenging year for research, I'm really pleased to report that most of our Cure Parkinson's funded research has been able to continue in some way and is now back up and running. That's it from me. So I'll stop sharing my screen and I'll hand you over to Helen.
Lea, thank you so much. Actually, there was a message that popped up in the Q&A from Kathy who's been leading the PD sensors work, who says that that's all back up and running and they're collecting data, which is fantastic. So
that's fantastic.
Brilliant that you that you were here. It's great to get a live update like that. So that's really, really fantastic news. Lea, thank you so much. I think you know, it was such a challenging year for researchers last year, and it's so encouraging to see how resourceful people have been to make sure that research continues, and also to really understand how important research is in terms of clinical outcomes for people. So ensuring that there is a rich and varied research pipeline ongoing feels very, very important. I'm now absolutely delighted to introduce Professor Annette Schrag from UCL. Some of you will have met Annette at our research update meeting, the last live one that we did, which was actually in 2019. Professor Schrag talked to Simon and I about how they've adapted the ADePT study. That's a little bit of a tongue twister, I'm afraid, and how that's now getting underway. And this is such an interesting study, and the way that they have embraced some of the technology developments and to be able to offer remote assessments has been hugely important in this study. So without further ado, Eros, I'm going to ask you to now be doing the sharing of the screen, if that's all right. So Professor Schrag, it's fantastic that you've been able to join us this afternoon. Thank you so much for sparing the time to tell us a little bit about the ADePT trial, which is a trial that the Cure Parkinson's is supporting. And we'd love to hear a bit more about the background and the research question that this is going to be addressing. So Simon, I'm going to hand over to you to ask the first couple of questions, and then we'll probably box it and box it a little bit as we go on.
So again, yeah, thank you very much for joining us this afternoon, Annette. Very kind of you. Can we start off firstly just by discussing what exactly is depression?
So depression is a symptom complex of which low mood is the most easily recognized, but not always very obvious. Other people may experience a loss of enjoyment or loss of pleasure in things they used to enjoy, or they may have prominent anxiety, which often goes together with depression, loss of concentration, or some more physical symptoms like loss of sleep or loss of concentration or loss of appetite.
So how prevalent is depression in Parkinson's?
So we know that depression is incredibly common in Parkinson's. At least a third of people with Parkinson's have depression or depressive symptoms at any one time. And this is something that we know can occur at any point in the disease course, whether it is right after diagnosis or later on. And in many people, it also occurs before the diagnosis is made, before motor symptoms occur. So we know it is an integral part of Parkinson's. Just like the motor symptoms, it is one of the most common non-motor features of Parkinson's.
So with that in mind, what led you to this research question?
So we have recognized that depressive symptoms are common in Parkinson's over the last 10 to 20 years. And there has been increasing research, but there's still a deficit in evidence on what the most important or effective treatment is for these symptoms, particularly because we don't know whether a depression in Parkinson's behaves and responds in the same way as it would in people without Parkinson's. And this is the background to this trial. We are studying two different medications compared to placebo and are looking at the effect of these medications on the symptoms of low mood, loss of enjoyment, and other symptoms, primarily over 8 weeks, which is when we would certainly expect to see a change in the majority of people, but also looking at longer term effect over a year.
How many people are going to be involved in the ADEPT-PD study?
So we're aiming to include 408 people with Parkinson's, and that of course will require that we recruit not just from one or a few sites, but from a number of different sites across the UK. So we have worked very hard to include sites all over the UK, in England, and as well as in Scotland.
So we invited you to talk about this study at our last research update meeting that we held in person, actually, which was in the autumn of 2019. And then, of course, we've had COVID in between. So you know, it would be fascinating to hear from you the impact that COVID had, because you were just at the start of the recruitment phase back in the very early part of 2020.
Yes, obviously COVID has been problematic in every possible way for people with Parkinson's and for trials. So we know that the symptoms that we're really concerned about of depression and anxiety have really rocketed in that time, certainly also in the general population, but definitely also and particularly in people with Parkinson's for a variety of reasons. In terms of the trial, clearly we've all been preoccupied with, in the NHS to concentrate on COVID, and many of us clinical staff have worked on the wards and ITU on the clinical side, but also research staff has been redeployed to work on the many COVID trials, which have been a huge success because of the support of the research staff in the NHS. But it has meant that other trials had to come to a halt. But the second thing is, of course, the first principle of research is not to harm, and therefore we stopped recruitment and face-to-face appointments as soon as COVID arrived. And this is now starting to be released because the numbers are so much lower. And also the research staff is coming back to other trials. So gradually things are starting up again.
That's so encouraging to hear. Have you had the chance to think about the trial design in this time, to adopt some more perhaps patient-centric, less site-intensive type of outcomes that may well support people's participation going forward?
Yes, so we put things in place to enable people to participate in the trial without leaving their home, really. And I think we are one of the very few trials with a drug that can be delivered completely remotely. Obviously, it is easier to do face-to-face assessments, but we now have approval from the ethics committee to do the assessments remotely through video consultations, and send questionnaires, and also send the medication directly to people's homes. And we do all the assessments remotely. We do have a number of add-on studies, including that include some blood tests. They're not absolutely necessary for the trial, but we're interested in people participating in these add-on studies in order to understand depression and anxiety in Parkinson's better and look at change over time. So we will make some further modifications, but the core trial can certainly be delivered completely remotely now.
That's fantastic. I was going to ask, with regards to the add-on studies, Cure Parkinson's is funding a sub-study of the larger ADEPT study. Can you tell us a bit more about that, please?
Yes, I'm really grateful to the Cure Parkinson's Trust and the supporters, because, as you know, nortriptyline, which is one of the two medications that we're studying, has been of intense interest in terms of its potential neuroprotective effects. So in preclinical studies, there is a lot of evidence to suggest that it may actually change the underlying mechanisms of Parkinson's and thereby delay the progression of the disease. But these data are purely from preclinical studies, from various strands of preclinical studies, but it hasn't been tested at all in clinical trials. And because nortriptyline is a drug that has been around for a long time, it is unlikely to be studied by a particular pharmaceutical company, but we're using the opportunity of doing this trial, which has the primary outcome of improving depressive symptoms and anxiety, to also look at motor progression and whether nortriptyline makes a difference to motor progression in Parkinson's over 1 year. And we've put a lot of things in place to try and look at that in more detail. And that includes assessments in the off period as well as in the on period. It includes an optional assessment using a sensor that people can wear on their wrist or on their body. And it includes a digital analysis of the assessments we do on video or in person. So these are optional add-ons. We're hoping that many people will participate in these, but they will help us to look in as much detail as possible in at the effect of nortriptyline in changing the progression of motor features.
That's fantastic. How are people going to get involved? Who's going to be the perfect candidate for you to get involved in this trial, Annette?
So we're trying to be as inclusive as we possibly can. Obviously, any drug trial has inclusion and exclusion criteria, and they relate particularly to potential side effects these medications might have. But in terms of the Parkinson's side, we're really interested to hear from anyone with Parkinson's at any stage. So it's not restricted to people at the very early or phase or with particular features other than having some features of depressive disorders such as low mood or loss of enjoyment or anxiety. We obviously have to recruit linked to hospitals, and as we progress in terms of opening up sites, more sites will become research active with this trial. And I know that you very kindly have a page on your website giving more detail on the study, and also with a link to our email to find out more about the study directly from us or from one of the sites. So we can direct people who are interested and give them more information.
And I just wanted to check, clearly you couldn't involve people with Parkinson's who are already taking an antidepressant of any form, shape or description.
Yes, absolutely. Occasionally people in clinical practice are on more than one antidepressant, but that's the exception. And because it is a trial, we can only include people who are not currently on an antidepressant. They can have been on an antidepressant before, but at the time of inclusion, and certainly several weeks before then, they cannot be on another antidepressant.
That's incredibly helpful. So in terms of just going back to the potential to do a remote delivery of a trial, one of the challenges I think that we face in Parkinson's clinical trials is that the outcomes are all based on clinical outcomes. And so this is a really novel approach to be able to deliver so much of your trial to its primary endpoint in this remote way. And what led you to embracing all this technology? Did you have all the technology in place at the start of COVID, or did you have to go shopping around to find the right resources to enable you to make these changes and adaptations?
It is something that we've always wanted to do from the beginning, but clearly with the COVID-related changes that the healthcare service had to do, this accelerated this, and it made it very clear that we have to have some more remote assessments. It is not as easy to do as with face-to-face assessments, but we believe that the digital tools will give us additional information. Clearly, these are research tools at the moment. They're not incorporated into clinical practice, so we still need the standard assessments with the scales that we use, but they will give us, provide us more information, with more information.
And Annette, finally, just one question. What is the time frame of the study? When will we learn the results?
So it all depends on the recruitment, and clearly this has been slower because of the circumstances. But we will be recruiting for certainly at least another year, a year and a half. And then people will be in the trial for a year in order to capture the change over time, for which the time frame of 8 weeks or even 6 months is not long enough. So it will take a few years until we have the results, because at the end, of course, we will also need to analyze it all. And up to that point, the blinding won't be broken, so we won't know before then. But luckily, we are now recruiting, and hopefully we will recruit fast.
Can I ask another question, just in terms of, you know, implementing this new approach to trial delivery, which is what COVID has triggered, I suppose, in a sense. Have you thought about trying to include some form of assessment for the healthcare professionals about how well it's working for them, but also how well it works for the participants? Because some participants may have been in other studies, and being able for them to be able to compare, even just in an informal way at the end of the trial, whether they found this beneficial, you know, which approach they've preferred, trying to understand the impact on the healthcare team. It could be really helpful for the field in terms of designing trials going forward into the future.
So it's not part of the actual trial itself, but we are involved with research studies to look at exactly that and to guide other trials of how to do this, because clearly this is a problem that a lot of different trials have had in the last few months or in the last year.
And I think we're all trying to learn about the best way forward that has the least impact on people living with Parkinson's to participate. But particularly, you know, I think it's such an interesting study, this, because you are seeking people with Parkinson's at any stage. You know, it is that inclusiveness that I think is so important. And too often we've heard from people with Parkinson's saying, "Oh, I'm too old, I'm too young, I'm too advanced, I'm too little into my journey," you know. So actually it's wonderful to have a study that really can involve as many people as possible, assuming they have the inclusion of low mood and the depressive types of symptoms.
Yes, I think it's a real opportunity also to open up a trial to a wider population, because traditionally research studies are in particular sites and they require relatively frequent visits. So there is a large number of people excluded from those trials, whereas with remote assessments or largely remote assessment, it becomes much more accessible to people to participate in trial if we can assess from wherever they are using these remote methods.
Annette, I really love this study because it's got so many interesting components, so many important components. You're dealing with a very important non-motor feature of Parkinson's and depression. You're doing a disease-modifying aspect as well, and you've got the remote assessment. These are all fantastic components. What have you learned as an investigator in the setting up of this trial? What do you wish you had known at the start, I suppose, is the better question. Now that you've been through the process of setting up the trial, what would have been useful information at the start?
I think one of the key issues has been that depression, as I mentioned in the beginning, is often undiagnosed, under-recognized, and therefore people who are eligible for the trial would not have been identified. And as soon as they have been identified, they are already taking an antidepressant and therefore not eligible anymore. So if I had to start again, I would make it easier from the beginning for people to identify themselves as potentially eligible and ask about the trial, because we can then always at the screening visit check whether they are or not, and then discuss with them whether if they can go into the trial, and if not, what other options there might be for their symptoms.
I wonder if you could just say in one short sentence what the study is.
The Adeptpd study is a trial with 2 medications, nortriptyline or escitalopram versus placebo, to improve symptoms of low mood, anxiety, loss of enjoyment, and other depressive features in people with Parkinson's. We are looking for 408 people with Parkinson's who may have these symptoms across the UK at any stage of Parkinson's.
Fantastic. Thank you very much indeed. Thank you so much, Annette. Annette is also involved in another study called ADePT-PD, which is to understand anxiety and depression in Parkinson's. And there are further details about both this and the ADEPT study on our website. So I'll put the link in the Q&A or in the chat in a little bit, so that hopefully everybody will be able to see that. Finally, I'd like to introduce Professor Anthony Schapira, who is also from UCL, and both Annette and Tony will be joining us for the Q&A session, which will start in about 25 minutes, I think. Professor Schapira has been leading a program of research to understand ambroxal in Parkinson's. He has a particular interest in GBA Parkinson's, and in this discussion, I was joined by our Dr. Joy Duffin, who's been leading the ambroxal research that we've been doing from our side. And so Joy and I had a wonderful discussion with Tony about this program of research. And then after that, Tony will be joining us live. But just as an aside on this, our trustees who met on Wednesday, I'm absolutely delighted to report that they have extended some funding to PD-Frontline, which Tony will be talking about a little bit in this talk coming up. And we'll be able to take some more questions around that, which is fantastic, with the vision that we're going to be able to try and recruit up to 3,000 people living with Parkinson's into this study. Without further ado, Eros, over to you to share your screen again. Thank you so much. Tony, thank you so much for joining us this morning. I wanted to start this discussion by asking you, why is GBA an important research target in Parkinson's?
Thank you, Helen. Well, I think for several reasons. Probably the most important is because an abnormality of the GBA biochemical pathway is probably the commonest association in the cause of Parkinson's disease. And let me just say what I mean by the GBA pathway. So GBA stands for glucose-3 bisidase, and it's a very important enzyme which is present throughout the body and takes part in the process of destruction of unwanted proteins and other parts of the cell, etc., in what are called lysosomes. Lysosomes are part of the cell that collect and destroy and get rid of all the rubbish. And that's, of course, very important because you don't want that building up in the cell. Also, the GBA enzyme, it participates in an important pathway in lipids, as we call them, or fats as they're sometimes otherwise referred to. And lipids are a really very important part of the function of cells. So first of all, the envelope that surrounds a cell and protects it is made up of lipid, as well as protein and other things. All those things inside a cell, like the lysosome, like the mitochondria and other bits, are made up of lipid as well in terms of their envelope. So lipids are very important. So GBA therefore clearly has an important role, not only in the turnover of things in the cell, but also in terms of maintaining the lipid integrity of the cell and its contents. And probably the best example of why lipids and the destruction are important in the context of GBA is what happens when you have very severe GBA deficiency. So this can be brought about by having a mutation in the GBA gene. And everybody has two genes for everything, one from the mother and one from the father. And if it just so happens that a person has inherited a faulty, or let's say mutated gene from both parents affecting GBA, GBA is very severely reduced in terms of its activity. And what happens then is we see a buildup of protein, lipids, etc., in cells in the body, enlargement of the liver, the spleen, and in some cases, very severe brain disorder as well. This is called Gaucher disease. So we can see that GBA plays an important role in the functions of the body and the cells. Now, the relationship to Parkinson's disease is very important because the same sort of mutation that, when present twice, causes this Gaucher disease, also, even when present in only one form, increases the risk for Parkinson's disease about 20 to 30 times. And what we find is that patients with Parkinson's disease, about 10 to 15% of them have abnormalities of the GBA gene and pathway. This makes GBA the most common association with Parkinson's disease in terms of a genetic cause. But what is even more interesting for those without a GBA mutation is that the GBA activity is reduced in the brain. And this is thought to come from an accumulation of alpha-synuclein, which, as people know, is really very important in the cause of Parkinson's disease. So one of the effects of alpha-synuclein is to reduce GBA activity. So for all of these reasons, the GBA pathway has become really probably a center of attention in Parkinson's disease research, particularly in terms of how can we manipulate it to slow down Parkinson's disease? How can we restore GBA activity when it's defective? So that's why I think GBA has become the center of attention for many researchers throughout the world, trying to understand the link between GBA and Parkinson's disease, and importantly, for many large pharmaceutical company and research corporations that are looking at the GBA pathway and thinking, how can we modify this to the benefit of those with Parkinson's disease?
Tony, having been very much converted to ambroxol as a fine candidate to intervene in the way that you have so eloquently described, can you explain why ambroxol? I mean, there are a lot of approaches in development, and one can see that one can target different aspects of this. But I know there's some very special features of ambroxol, and I'd be very grateful if you could expand on that for us.
Yes, of course, Joy, thank you. So perhaps if I give a perspective of how people are thinking about the GBA pathway and manipulating it in general, and then talk about ambroxol specifically. So people are very excited about this GBA story for the reasons I've just stated, that there are many ways in which it might be manipulated. One of the main targets is to directly increase GBA activity itself. So restoring, if you like, the normal activity of the enzyme to those with a GBA mutation, but also to those without a GBA mutation even, because as I've mentioned, the buildup of alpha-synuclein causes this secondary reduction in GBA activity. So people have been looking at this in many different ways. And one way is to identify what we call small molecules or chaperones, which can target the GBA defective enzyme and restore its structure and activity. And another way is to actually increase the activity of the GBA gene, usually by linking the GBA gene to something like what we call a viral vector, and then inserting that into the brain to increase the expression of the GBA gene and thereby increase the activity. That usually involves an injection of the gene linked to the virus, either directly into the brain or into the spinal fluid. Now, those techniques and using AAV or the virus link to the GBA gene are under development at present in what we call phase 1, and some are entering phase 2 studies. So first of all, to try and prove safety, and then to try and prove that you're having the desired effect in principle. Another way was to say, well, the GBA gene and enzyme are not functioning properly. And this might lead to the buildup of what we call a substrate. A substrate is what the GBA enzyme destroys. And when it's defective, that substrate will build up. And this is what you see in Gaucher disease. Now, we never actually saw that in Parkinson's disease patients, but it was a perfectly reasonable idea that this buildup of the substrate, as we call it, might be contributing to Parkinson's disease. And this was the basis behind the recently finished Venglustat study, because Venglustat reduced that substrate buildup, but didn't actually affect the GBA activity itself. It was worth a try, and it was a very carefully undertaken study, but it failed in terms of showing any improvement for Parkinson's disease. So I think that the idea of increasing the GBA activity itself is really the main target and the most exciting approach. And that's where ambroxol comes in. So ambroxol is one of those chaperones that I mentioned earlier, which means, as the name implies, that it is able to link onto the abnormal or defective GBA enzyme and chaperone it through the cell to where it should work in the lysosome. And in addition to that chaperoning activity, it also has an interesting effect on generally increasing the activity of this lysosomal pathway, which helps in the destruction of all those unwanted products in the cell. And it probably, as a bystander effect, if you will, of increasing GBA activity, restores the normal lipid profile of the cell. Now, this, I think, is the most exciting and most logical approach to the GBA pathway treatment. And as I mentioned, that's where ambroxol comes in. And fortunately, you don't have to inject ambroxol into the brain or into the spinal fluid. You can take it as a tablet. And our first ambroxol study showed that people can take ambroxol by mouth. It's absorbed well into the bloodstream, and then well absorbed from the bloodstream into the spinal fluid and by presumption, therefore, into the brain. It's very well tolerated. It's been around for a long time, so we know a lot about ambroxol. It has been introduced as a cough linctus many decades ago and still used as such. So we know a lot about it. It's safe, it's effective in terms of increasing the GBA activity in cells models and in animal models. And importantly, in our cell models derived from patients with GBA mutations and in our mouse models, the ambroxol has been able not only to increase the GBA activity, but also to reduce alpha-synuclein levels in those models. And there's the key. So it's doing what we want it to do in all of these different models. What we want to know now is it capable of doing it in the Parkinson patients?
That explains a lot of the background thinking that has brought us to this point, Tony, and it would be very helpful now to hear a bit more about the study plans that we've been working on for a long time now and that are at an exciting point of being very close to progressing, and what has led to the plans that we have in mind, some of the discussions and so on. I wonder if you could expand a bit on that.
Yes, thank you, Joy. So our present thoughts and design on the development of ambroxol builds on the first trial that we did, which is a small study, what we call a proof of principle, if you like, and we named it AIM-PD. And the principle of this was to confirm that ambroxol taken by mouth gets into the brain and reaches its target and increases the GBA protein. And it proved all of that. And it also at the same time proved that it was well tolerated and safe over the 6-month period that patients took it for, and in the sort of patient group that we're targeting, those with Parkinson's disease. So that small study was really important as the foundation for our thinking for the next study. And the next study is seeking to answer the question, well, of all these things that ambroxol does, can it actually slow down the progress of Parkinson's disease? Not an easy question to answer. So we've been thinking a lot about this, talking to a lot of people around the world, getting their advice on it, and importantly, getting the advice of the NIHR here in the UK that is responsible for giving a license to drugs so that they can be prescribed. So in trying to look at the design of this study, we thought, well, the sort of people that we want to test are those with Parkinson's disease, and at the same time looking at those with a mutation of the GBA gene that, as I mentioned before, is the commonest association of Parkinson's disease, but also those without the GBA gene, but with Parkinson's disease. And that's because we think that ambroxol may be of benefit to both those with and those without a GBA mutation, and it's worth testing that right at the beginning. So we're going to be looking at quite a large number of patients. I think the number is somewhere around 165 patients with Parkinson's disease. And we're going to be giving them ambroxol, but at the same time, we're going to be giving an equal number a placebo, because it's essential in order to understand if ambroxol works to compare it to the placebo. And in terms of how will we know if it's slowed down Parkinson's disease, we're taking what is a fairly standard approach, looking at a well-established rating scale for Parkinson's disease that looks at how the patient feels and how the doctor interprets the changes of the person with Parkinson's disease on clinical assessment. So it's a combination of both. And that I think is what is really important. It's all very well for a doctor to think that a patient is doing well, but actually if the patient doesn't feel that way, it doesn't really matter. So it's really important that the scales that we've used test whether the patient themselves feel that ambroxol is helping them. And we're going to run the study over 18 months and then make the assessments during that period. And we're going to be doing a number of assessments, clinical and some blood tests and so on. And then at the end of 18 months, we're going to have a 6-month period where everyone in the study will be given ambroxol at the dose of 1,260 milligrams a day. And that 6-month period at the end of the main study will be important in seeing how all of the patients respond to ambroxol over this period. So those are the sort of basics of the trial design. And as I say, we've carefully worked on this and spoken to the MHRA, listened to their advice on what they would be looking for if the trial was positive, in order to think about whether they can give a license on this trial's results, or alternatively, whether they think the results are encouraging, but we need to do more. That's become a very important influence on the design of our study.
So I've got a question actually, which is for both and you, Tony, next, which is around the changes that we've introduced for the drug between ADePT-PD and the proposed next study. Because in ADePT-PD, I understand that people were taking 21 pills a day, which is quite a big pill burden, and particularly as they didn't taste very nice. So I wondered how you two solved this problem.
Well, let me just start by saying a few words about the problem and then hand over to Joy, who found the solution. So the problem, as you quite rightly said, was in our first study, ambroxol was only made in 60 milligram tablets at its highest strength, because it was used as a cough linctus, and that dose worked for coughs. But we thought we needed much more to work on Parkinson's disease. In fact, 21 tablets a day, so 7 tablets 3 times a day, is what the patients had to take, and they were heroic in taking those. But as you say, didn't like it that much, but persisted with it. So we had to find a solution to make it more acceptable to patients in a large study, so that they would take the dose that was required. And that's where Joy came in and found a solution. So Joy, you mentioned a story.
Well, thank you for the credit. That's not entirely true, but we'll get to that. So I certainly enjoyed the challenge, because although I had a little bit of a chemistry background, I started to explore the whole aspect of reformulation, and in particular, ambroxol, its particular characteristics that might present a challenge in making a more concentrated tablet. And through various references and guidance and recommendations, we spoke at greater length with a couple of rather specialist producers that could help with this. And they came up with a couple of proposals to look at what happens if you try to concentrate it, because we understood it might be a bit of a problem. It's not particularly soluble in water, which is quite a good thing because it helps it get through the blood-brain barrier, as I understand it, Tony. But it's not good when you want to concentrate it and it goes into a kind of gel and you want to produce a solid tablet. However, although our discussions with 2 very, very positive and helpful groups that have helped cure Parkinson's in the past, and I'm sure will again, went very well. It's actually thanks to Tony, because of his reputation and interest and champion and knowledge in this subject, that he was contacted by a group in Norway that were working on ambroxol and had an interest in a similar area. And when we spoke to this group, thanks to Tony's introduction, we found that they had done quite a lot of this background reformulation work, and it had proved, in fact, to be slightly less challenging than we'd expected. And it does look as though a solution is there, and that one can produce quite a concentrated tablet without any major changes, which was one of our concerns, because that would have affected its whole safety record and its regulatory aspects. But it looks as though we can produce well, the Norwegians can produce a concentrated tablet such that the dose that we think will be required is now reduced to 1 tablet 3 times a day. And I've seen them, and they're not too big, and they're still a little bit chalky. But for instance, compared to the other thing that we were looking at, which is that there is a syrup that's produced for cough medications. And I did acquire some of that as well and had a little taste. And it was fine, but it's still a very substantial wine glass of syrup 3 times a day with a lot of sugar. So you know, probably not ideal. I think the feeling is all round, and we did take some consultation with some of our prospective participants and those that had previously heroically taken the tablets before, and the feeling was that this tablet solution from Norway will be the optimum way forward. And we are very pleased to have had that introduction. And I say that is thanks to Tony's involvement and status in this field. But I will take a little bit of the credit for having kept it going along the way.
So it really does sound as if this last year has been a year of problem solving in terms of making approaches to the MHRA and understanding their thinking on how we could move this forward, so that if the next study is successful, we can move forward with a license, understanding some of the challenges around patient burden with the drug, and then reformulating and identifying a source of medication that actually would be robust and safe and not changed in any way, but just in the number of pills that people have got to take, which has got to be an important step. So I think my next question really for Tony is, who is the perfect participant in this study to come? And how could people help now?
So the people that we're going to look to recruit for the next ambroxol study, obviously we'll have Parkinson's disease confirmed by their physician. They will be relatively early in the course of Parkinson's disease, but not right at the beginning. So patients may or may not be taking therapy, symptomatic therapy for their Parkinson's disease. And they will, of course, be allowed to continue that through the trial. And if it becomes necessary for them to increase the symptomatic therapy on the recommendation of their doctor, then that will be possible too. So the perfect patient has been confirmed to have Parkinson's disease, be sort of in the early to mid stage of Parkinson's disease, and able to travel to their local Parkinson's disease site that we will be setting up around the UK, understanding the nature of the study, able to consent to it, etc., able to take the tablets, and so on, have some blood taken. And because it is really important for us to be able to confirm the ambroxol is having the desired effect on the processes of Parkinson's disease, we will be looking to ask the people that come into the trial to have a lumbar puncture at the beginning and the end of the study 18 months later. And this is a really important part of all of the study, so that we can prove that the drug is getting to where it needs to work. So the perfect patient has all of those features, and of course, men or women, they can be any age up to, I think we said 75 in the study. and of course willing to stick with us for that 18-month period and then the 6-month period that follows. So a total of 2 years for the study.
So Tony, how can people put their hand up now? How can people get involved now? Right,
so I think we have set up, with the help of the Cure Parkinson Trust, a website called Frontline PD. And the sort of catchphrase is get trial ready. And the benefit of Frontline PD is it will take everybody who has a diagnosis of Parkinson's disease and recruit them into this Frontline PD. And part of what we do is to check the DNA to see if they have a mutation of the GBA gene. We're also looking at another gene called LRRK2, but the GBA gene. So we will be able to say to the patient if they wish, you do or you do not have a change in your GBA gene. And that is really critically important to be able to then consider entering the new Ambroxal study, because as I mentioned, there are going to be 165 people with and 165 people without a GBA gene change. So we're looking for a lot of people. And the way to get considered for the study is to log into the PD-Frontline website. It's very easy to do, very easy to follow. And there are people that we have here to help patients, people, carers run through the website, but it is terribly simple. And then we just send out a simple saliva kit. And it's just like a swab in the mouth. And people post that back to us, and we do the rest. So that, I think, is probably one of the main ways in which people can get linked into the system for entering not just the new Ambroxal study, but other new trials as well that may come up with other things that target not just the GBA pathway, but other factors too. So I think it's a really good way for people to get in contact with all of that process. It doesn't mean that you have to do any of the trial work. It just because you go into Frontline and sign up for that, it doesn't mean that you have to then enter a trial. It's entirely up to you if you do or not. And you may want to go into one trial and not the other, and that, or none at all. It's entirely up to the patient. But that PD Frontline, I think, is the best first step for people to consider.
Annie, that's fantastic. And I think particularly we found that last year, it was a real success of lockdown. It was one of the few studies that people could still continue to take part in. Oh, sorry, I've got a small dog who's barking at the same time. And I think it's a real achievement to have kept that study online and been able to recruit as we've gone on. Tony, thank you so much. I am going to draw this to a conclusion. A small dog is really going to get overexcited at this moment. But thank you so much. That was very, very informative. And I think you've given us a huge amount of insight into the complexities of this program, but also, I think, importantly, how this has been a year where we've solved some of the problems that we were facing with this as a program of research. And we do continue to see it very much as a program of research. So thank you very much indeed.
Oh, thank you, Helen, and thank you, Joy, and thanks to all of those that work with the Cure Parkinson Trust to make all of this possible, because without your help, frankly, it wouldn't be possible. So thank you.
So thank you very much, Tony, and thank you very much, Annette, again. I'm now going to hand over to Simon Stott, who is our Deputy Director of Research, if you haven't met Simon. And Simon's going to lead us through the Q&A. Actually, it'd be wonderful if Tony and Annette, you could turn your cameras on, and also Richard and Joy as well. That would be fantastic. But Simon, over to you. And thank you very much. Do keep your questions coming in through the Q&A. I know Simon's been grabbing some as he's been going and answering some as well. Simon, over to you.
Thank you very much, Helen. And thank you everybody for attending today. I'd like to start off this Q&A session with a broad question for all of the panelists. There's been a lot of discussion in the presentations about COVID and the impact that it's had previously, past tense, on Parkinson's research. But I was wanting to get folks' opinions as to what they think some of the challenges going forward are going to be in the post-COVID world. And more importantly, what folks who are watching this event today could do to help. So perhaps I could start with Annette. You're on mute, sorry.
The usual mistake. Thank you very much for asking me to be here and answer questions also on the ADEPT-PD trial. So COVID obviously has been a huge challenge for any studies recruiting patients into trials because of the risk attached to going to hospitals for assessments. One of the things that we have also done, like Tony just described, is go online as much as possible. And obviously that is not for everyone, but a lot of people have indicated that they do want to do things online. So going also to our website and engaging with the sites that are already open, that are on the website, would be very helpful to help us recruit for the trial, because we have started now for the trial and there are more and more sites coming on board.
And Professor Schapira.
Thanks, Simon. Well, apart from all of the tragedy associated with COVID, there have been some interesting developments and opportunities, and I think changes in people's behavior as a consequence. And they have become generally much more comfortable with being on the internet, using their computer for different things, including, for instance, this type of video meeting. So I think there has been a shift in behavior which has made something, like Annette says, easier, such as going online and doing studies. But when it comes to clinical trials, of course, in-person attendance is necessary for interventional studies. So for instance, I know you heard a lot about the Ambroxal study, but for that, people will have to come to the different centers that would be looking at this. I think as we move forward, particularly with the very successful vaccination rollout, the changes that have been introduced in hospitals, and the way in which we can manage patients who come to hospital, things are much safer. And hopefully people will feel more comfortable with attending hospitals, both themselves and their carers if necessary. And I think the safety measures put in place will likely still be present, but probably as we move yet further forward, less and less intrusive and less necessary. So I'm hoping that as we move into 2022, clinical trial work will get back to normal.
Lea, can I ask the same question of you with regards to some of the preclinical work, etc.? What are some of the potential challenges going forward in this post-COVID world, and how can folks who are watching this event today help out?
So with the preclinical research, it's been a bit different in that research has been able to continue, but maybe at a bit of a slower pace, particularly because most universities opened up again in the summer of 2020, but had to put in place COVID secure measures, which might mean only having certain numbers of people in the labs or certain working hours within the labs. So things are going a bit more slowly, but the work is still going on. And I think getting involved with some of the preclinical researchers is always a good idea, working on potentially new grant applications with the researchers, to always keep a focus on people with Parkinson's when we're thinking about the preclinical research and how it can potentially then lead on to clinical research.
Thank you very much. I'm going to shift now to the specific, some of the specific studies we talked about today. And then, Annette, I'd like to come back to you here. One of the questions from one of the attendees is, do we know exactly, or do we have ideas as to how depression is caused in Parkinson's? Could it be medication related, or is it more the anxiety and the impact of realizing that you have a condition like Parkinson's? Or is it a chemical imbalance in the brain? Or is it all of the above?
Yes, so depression clearly is not just one very uniform disease. It's a syndrome. And even outside of Parkinson's, there are multiple factors that range from a genetic predisposition, people with a family history of depression, to life events, personality characteristics, and of course having a chronic physical disease is also other physical diseases are associated with depression. And that's why it used to be thought that depression is just a reaction or is no more common than in the general population. But that's actually been clear now for 1 or 2 decades that it is more common in Parkinson's and an integral part of Parkinson's itself. And we know that it can start before the motor symptoms and is more common in people who later get a diagnosis of Parkinson's. So it clearly is a reflection of the ongoing disease process and not just a reaction. Then the question is, which aspect of the disease process is responsible for it? And we do know that the dopaminergic deficit that is very prominent in Parkinson's is at least partly responsible for it, because often people have, when they have an off period, they experience low mood, anxiety, or even panic attacks, and that lifts with taking medication. Clearly not in everyone, and in many people it isn't clearly related to fluctuations, but that very much suggests that dopamine is involved in that. And there are some imaging studies that also support a role for dopamine, but there are also other brain metabolites that are also involved. And depression in general is often treated with SSRIs, which clearly address other metabolite deficiencies. So it probably is a mixture, and it's not exactly the same in every individual.
And for folks who are interested in potentially getting involved in the ADePT-PD trial, this is going to be recruiting 408 participants nationwide. There is a website, I understand, a webpage on the UCL website with a suitability quiz. Are you able to talk more to that, please?
Yes, in fact, on this website, which I know you're also linking to on your website, there is a little bit of information on the trial, just outlining it. And it also gives our contact details as well as contact details of the sites that have already opened. And this is an ongoing process. So we will hopefully have 30 sites across the UK, and a handful are open now and others are coming on. But clearly a lot of the sites are struggling with staff capacity because of the other ongoing studies on COVID and other studies. But that there will be an increasing number, hopefully covering all of the UK. And because the core of the study can be done completely online, we can even do this when people live quite a long way away. So if you have any questions, then please do contact us.
Did we just lose Simon by any chance? Oh, Simon, you briefly for a moment. Simon had a bit of an internet issue earlier today. Oh, golly. Oh, for a face-to-face meeting, it'd be so much easier. Back to you, Simon. Are you frozen again, Simon? I think you might be frozen. Oh, I'm not sure. In which case, I'm going to pick up the lead here. And Simon, just unmute us and when you're ready. Tony, we've had a question come through on the live Q&A, which asked, you mentioned the role of GCase in the lysosome. Is there a way to actually measure GCase activity in the lysosome as opposed to measuring GCase in the spinal fluid?
Very interesting question. So measuring GCase specifically in the lysosome is exceedingly difficult to isolate pure lysosomes. We are actually actively working on that to isolate lysosomes from white blood cells that circulate in the blood, but it's exceedingly difficult to get enough material. But one can measure whole cell GCase activity, which isn't as good as isolated lysosomes, but it's still nevertheless a reasonably good indicator. And actually, the CSF GCase activity correlates very well with the blood GCase activity.
And Tony, there's another question for you as well. Will DBS be an exclusion criteria for Ambroxal in the future as we get towards that?
Yes, it likely will be, because we will be recruiting people probably at a stage rather earlier than DBS would have been required.
Okay. And there is another fantastic question that also came from an anonymous attendee. So if you bear with me 1 second, I'd be very keen to get this one answered. Even for those with a GBA mutation, there can be a wide range of GCase levels. Has there been any consideration to only including those subjects with an abnormally low level of GCase?
Yes, indeed, a very good question. And we have given this much thought. But I think that there are two issues. One is that the proportion of people with very low GCase activity is very small, and we probably wouldn't be able to identify sufficient numbers. But the second thing is we think it's probably not essential to have such low GCase. It's not just the low GCase activity that causes or is associated with the Parkinson's. We think it's also something else. Hence, a very good question, but we haven't selected just for those with very low activity.
And another question for you, Annette. What was the rationale for selecting specifically nortriptyline and escitalopram as compared to others?
So it's a very good question, and we discussed the choice of the respective SSRI and tricyclic carefully, because the grant is in response to a call from the NIHR, which specifically was about identifying whether tricyclics or SSRIs are better for treatment of depression in Parkinson's, because that's where the gap is in evidence. And nortriptyline, and one of the reasons why I was particularly interested in this call is because of the suggestion that nortriptyline might have some disease-modifying properties in Parkinson's disease. This evidence isn't there for other tricyclics, so this is why we're particularly interested in the nortriptyline arm. And sorry, just to talk about escitalopram, it's a newer and better tolerated version of citalopram, which has been around for a long time and is off patent and now widely used. So one of the newer and most effective SSRIs.
Thank you very much. Simon, back over to you. I'm putting you back into the hot seat. We're due to finish at 2 o'clock, and there was one rather wonderful question about the 10 to 15 years, which I wondered whether you might be able to fire.
Yeah, so I was going to throw this at Richard. I hope my internet doesn't cut out before I do. Richard, I was wondering, one of the panelists, Simon, has asked with regards to all the fantastic research that's going on at the moment. There's always this sense of we're improving understanding and management of the condition, but how far away are we from sort of seeing real sort of disease modification?
Well, that's a question I've heard for the 14 years I've been in Parkinson's research, but I think that we've made great strides in the last 2 or 3 years. I think we've found a couple of our results of our trials and other related trials have shown some evidence of disease modification. There's a publication due, which will show another one. They don't work in all Parkinson's patients. So part of our job now is to decide which patients will respond to those therapies and make sure the right patients finish up on the right therapies. So, the trial done by Tony Shapiro's colleague and Annette's colleague at UCH on exenatide was published in The Lancet 2 or 3 years ago and showed substantial benefit in some patients and no benefit in the others. And overall, it was beneficial to the whole group, which has led to a phase 3 trial in that drug, which is ongoing. And of course, Tony Shapiro is going to do a phase 3 trial in ambroxol. But also the calcium channel blockers have shown considerable benefit, but not yet published. So, look, I think we've got new compounds coming through with great promise, sort of compelling logic to test them in Parkinson's disease with an expectation they will make a benefit, but also the drugs that we've got already into trial, some completed trials have shown huge promise. And I think that we will be identifying which patients respond to which drugs and sculpturing benefit across a very large population of Parkinson's patients who will respond differently amongst themselves. And our job is to work out who's going to respond to which drug.
Wonderful. Alan, how are we doing for time?
I think at this moment, we need to bring this meeting to a conclusion, but I particularly want to thank Camille, who's managed to join us from her NIHR specialty meeting for a brief 2 minutes, but also Tony, particularly thank you to you and Annette for joining us today. And thank you so much for sparing time to allow us to talk to you beforehand. I have to say, it's never straightforward doing these meetings via Zoom. And I really want to thank everybody who's joined us today, but also for really interesting questions that have been coming through. There are a number of questions we haven't been able to get to. We will do our very best to reply to those offline and contact you individually. But once again, thank you all so much for being here. And thank you so much for fascinating talks. Without further ado, I'm going to say goodbye. Nice to see you all.